Muy bien. Buenos días, buenas tardes. Today, our invited speaker is Dr. Vijaykumar Muley. As already you know, he is an associate researcher in Dr. Alfredo Varela's laboratory. I'm going to tell you some aspects of his academic career. He studied in India, biotechnology first, and then he specialized in bioinformatics. He did a master and PhD studies in this topic of bioinformatics. Indeed, he is an expert in this field of research. Before Mexico, he was working. He joined laboratories in Europe, first in Germany, and then in Ireland. And in 2 years ago, he arrived to Mexico to join Dr. Alfredo Varela laboratory. As I said, he is an expert in bioinformatics. He is first author of a very interesting paper published last year in Progress in Neurobiology. And today he's going to tell us about a very interesting project also in transcriptomic analysis of SARS-CoV-2 entry receptors. It's very a hot topic in these pandemic days. Thank you very much, Vijay, and we are listening to you.
Thank you very much, Mauricio, for your kind introduction and also for invitation to talk about my work on COVID. So just to mention, this is not my my primary work. I work on neurodegenerative diseases, but since last 1 year, I have been working on this topic. So whenever I get time, I do this work, and after 1 year, I think this work is now in good shape. So today I'm going to talk about two membrane receptors of human, which SARS-CoV-2 elegantly used to develop COVID coronavirus disease 19. When I started this work, it was not idea to find actually potential targets. So that time nothing was known about this virus, and I just wanted to know what is the reason behind its high infectivity and how this virus can escape from China in 2019 December, and within 4 months it's already spread in more than 200 countries. And then I started this work by doing some transcriptomic analysis of these cell entry receptors, and then I found some genes which could be really interesting to study further. And since this work was purely computational in nature, I wanted to confirm whether the findings of findings in mouse also can be confirmed in mouse. So that's the reason the title of this talk is cross-species transcriptomic analysis of SARS-CoV-2 cell entry receptors reveal potential therapeutic target genes in the human genome. So coronaviruses are zoonotic pathogens. It means they are originally adapted to non-human animal host in which they usually don't cause diseases. But sometimes these viruses, they come in contact with human directly or through an intermediate host and can cause disease. And mostly these kind of diseases actually catch us in surprise because we don't know these viruses and we don't know vaccines or any treatment. So there are, so coronavirus is actually a large family of viruses. They are divided into alpha, beta, and gamma coronaviruses. So gamma coronaviruses, they infect only birds. alpha and beta, they infect humans. So we know these viruses since 1960s. There was the outbreak and outbreak of flu-like respiratory illness, and people, researchers, two independent researchers isolated two viral strains. So initially, it was assumed that these were closely related to a virus, infectious bronchitis virus, which was found which was actually caused outbreak in 1920s in domesticated chicken in north part of America. So when they compared these structures, they realized that these two viruses, okay, they are closely related to the virus which was found to infect birds in 1920s. And these viruses, they were distinct from influenza virus because they have spikes like projections on their surfaces. Okay? So there were 8 virologists. They wrote informal letters to Nature Journal requesting that these viruses should be named as a coronavirus family because they have distinct morphology from influenza virus. So after 60s, first outbreak was severe acute respiratory syndrome coronavirus 1, which emerged in 2003 June in south part of China, and this virus very quickly escaped from China and spread it in more than 25 countries. And it caused approximately 9,000 cases, and case fatality rate was 10%. Okay. And subsequently, people started to work on coronavirus again, and they found 2 more species. So it means these viruses actually are already circulating, already adapted to human population. So there was striking emergence of atypical pneumonia in Saudi Arabia. And now we call this virus Middle East respiratory syndrome, coronavirus. And this outbreak was kind of deadly because the virus was not as infectious as SARS-CoV-2, but the case to fatality rate was more than 35%. Okay. Of course, cases were less, and this virus is still circulating in human population. And even as of now, there are some cases emerge once in a while. So the SARS-CoV-2, now this virus was entirely different, and we have seen this in almost 1.5 years. It is highly infectious, but the case fatality rate, as I calculated, it was just 2.3. There are also reports says that the case to fatality rate is 2.5 or 3. But even then, it seems like case to fatality rate is not the problem. The problem is R0 number, which means how effectively it can infect humans, okay? And that rate is quite high. So since these are zoonotic parasites or pathogens, people started to identify their sources. Okay, and most of these sources they goes back to bats. Okay? Many people think, many researchers actually think, or they have already published a lot of literature on SARS-CoV-2 was transmitted from bats to civets or raccoon dogs. And from then it transmitted to human, but there is now a lot of evidences at experimental level that it is not the case. SARS-CoV-1 and SARS-CoV-2 more likely directly transmitted from bats to human, and maybe at the places where people sell exotic meat. So bats are actually very abundant mammalian species. Okay? And they not only accommodate coronaviruses, but they also host a lot of different viral families. Okay? And they don't cause any disease in bats. But what is surprising, the surprise which was come after 2003 is that the different bat species, they host more than 3,700 coronavirus species, and large part of this coronavirus, or in general virus diversity, we have no idea. So essentially, coronavirus outbreaks, there are chances that, considering the number of viruses bats host, and people use them as an exotic food, these viruses have the potential to enter in human population, like both SARS-CoV and MERS did. So this is just an overview what is happening when virus enters into our body. So so any foreign particles, when they enter in our body by any means, we have very good immune system which recognizes these foreign entities, virus or bacteria or any fungal pathogen, and generally the immune system able to neutralize them. So when I say neutralize, it's literally they choke them up from the body. But viruses also evolved with different mechanisms to treat this immune response and cause infections. So viruses are actually bulky. So they are made up of RNA proteins. So they are bulky in size. Okay? So they cannot simply just enter into cells, because host cells are actually safe for them to hide from immune response. So viruses have evolved with a lot of different mechanisms. So one of the mechanisms is like they have surface proteins. Okay? So they recognize specific receptors on animal membrane. Okay? And usually these receptors, they are involved in bulk transport, export or import of molecules inside or outside cells. Okay, so when these viruses with the receptors, they bind to the host membrane receptors, usually conformational changes happens, either host receptor or virus surface protein gets activated, and that leads to fusion of virus in the host membrane. Okay? And in this bird or cavity like portion, it pinches towards cytoplasm, and then the host membrane envelopes the virus, and then it can translocate to the cytoplasm. Once virus is in cytoplasm, it injects its genome and hijack cellular machinery to synthesize, to replicate its genome, and also synthesize protein which are required for its functions. Okay, so once all proteins are and genomes made, these viruses, they either go through exocytosis outside the cell, or they burst, and the cell will burst, and this virus release from the cells. So then the newly synthesized, newly produced viruses, they infect neighboring cells. So initially, this is local propagation of viruses, but sometime it also develops into clinical symptoms and actual disease. So in SARS-CoV-2, it has a spike protein which actually helps this virus to enter host cell and eventually develop COVID-19 disease. So the progression of SARS-CoV-2 can be divided into 4 stages. So after initial infection, or let's say cell is inside cell, host cell, first 5 to 6 days, we don't see any symptoms because the virus is locally propagating, but that propagation is not strong enough to actually activate immune response. And when you activate activate immune response, then we find we see the clinical symptoms. So nothing happens in case of SARS-CoV-2 in first 5 to 6 days. And then the viral load, once it is increases, according to literature, it seems like the viral load first increases in our nasal cavity. And then the virus starts propagating and migrating towards lower respiratory tract. And in 80% of cases, it stays in the upper respiratory tract and causes mild to moderate disease. So these individuals, they can recover without any medicine because of strong immune system. But in many individuals, this can develop some mild disease. But unfortunately, 20% of COVID patients, they develop severe to critical illness. Okay, and if you look at the features of these individuals, most of them are old age and with some comorbidities or preconditions. Okay, and these are susceptible for severe or critical illness, and the survival rate is approximately like 50% in these patients. Okay. So the most common complications that we have been seeing in this COVID disease is mostly atypical pneumonia, acute respiratory distress syndrome, acute liver injury, or cardiac arrest, and in many patients we see blood clotting. So this is a structure of coronavirus. So this is enveloped virus. Inside it has single-stranded RNA molecule as its genome, and then these are the spike proteins. So they form trimers, and then they project on the viral surface. Okay. And they appear as a crone, so that's why they are called coronaviruses, because crone in Latin is corona. And this spike protein has 2 subunits. One is S1 and other is S2. So S1 subunit, this virus uses to bind to its host receptor. Okay. And S2 subunit helps this virus to fuse into membrane and enter into cell. So SARS-CoV-2 employ mainly ACE2 and TMPRSS2 membrane receptor to enter into cell. But there are some alternative receptors also have been found, and also in some cases only ACE2 or TMPRSS2 with other proteins can also allow its entry. And that could be one of the reasons it is very successful in infecting. It's high infection rate. So what happens is, like this ACE2 and TMPRSS2, they are present on membranes of a lot of cells, actually, in different tissues. Okay. So first, S1 subunit binds to ACE2, And then the TMPRSS2, which is in close vicinity, it can bind to S2 subunit. And this interaction leads to conformational changes in TMPRSS2, and it becomes active, and it cleaves this S1 subunit from the coronavirus. Okay? And this complex TMPRSS2 and the coronavirus, it will be taken up by cell through endocytosis. So it is essential that these host membrane receptors more, not essential, but more likely to be involved in protein trafficking of large molecules inside our cells. So instead of looking for other alternative receptors, which have been shown like occasionally used, but ACE2 and TMPRSS2, not only SARS-CoV and CoV-2 we use, but also SARS-CoV-1 and also some other viruses, they use these receptors. So what I wanted to know is which genes are associated with ACE2 and TMPRSS2. So basically, which genes which other genes in human genome they can work with these two receptors. Long time back, I have worked on different methods which can predict protein-protein interaction networks at genome scale. And what I have found is the gene expression similarity between two genes in multiple conditions is often a good indicator of their functional relationship, or they are often part of protein complexes. So when we identify such co-expressed gene, it is more likely they work in same biological processes. Okay? And the reason for their co-expression is often because of the common regulatory mechanisms which are responsible for their inductions. Okay? And I have also shown co-expression of genes can be used to reconstruct biological pathways with high accuracy. And that is the idea. Use expression patterns of ACE2 and TMPRSS2 in multiple situations and identify which genes are required at that time. And more likely they are working with these receptors. So to do this, I did a large scale screening of microarray data sets. So I collected microarray data sets from approximately 1,000 independent published studies from GEO database. Those microarray data sets were contained more than 35,000 samples and covering expression of 80,000 genes, which is actually a number close to all genes encoded by human. And then co-expression of ACE2 and TMPRSS2 with all remaining genes was accessed using Pearson correlation tests in each of the microarray data set. And then I asked question, how many times I find significant Pearson correlation coefficient between ACE2 or TMPRSS2. So I found, so basically I give cutoff. Okay, so if I find ACE2 expression in more than 150 data sets out of 1,000, this number is less likely to be occurring by chance. So I selected 220 genes which were associated, which were co-expressed with ACE2, 344 genes which were co-expressed with TMPRSS2, and approximately 1,000 with both of these genes. Okay? And this was actually 1,255 genes. And then I wanted to see if these, let's imagine the virus is employing ACE2 and TMPRSS2 receptors for infection. It means that the downstream processing, normal downstream processing of processes of these receptors must be affected. So if I check data set where I have gene expression profiles of all genes, which are infected by virus, coronavirus or coronaviruses, and the mock experiment. Then I should be able to see what is happening to these genes after coronavirus infection. So I collected three different data sets from MERS-CoV, SARS-CoV-1, and SARS-CoV-2. And I then divided genes, which I identified, and the remaining genes. And I checked their expression. So it's very clear with all these infections, these genes, they are downregulated after infection. In fact, SARS-CoV and SARS coronaviruses, they actually use ACE2 and TMPRSS2 receptors, but MERS uses different receptor. Even then, I see downregulation of these genes. So it seems that all these genes may be involved one way or other way in viral infections. So next question I asked like what functions these genes are doing in cell. So I'm sorry, this figure is very big, but I have just noted down few some interesting terms which processes in which most of these genes were enriched. So it is found that ACE2 is mainly linked to genes which are involved in hormone response, innate immune response, viral processes, and wound healing. And this wound healing is interesting because if the deficiency of ACE2 in lung causes delay in repair of lung injury, and then TMPRSS2 again this has very unique functions. And what was striking for me was there were 73 genes out of 331 are involved in regulation of RNA biosynthesis. Okay, so it seems like TMPRSS2 works with genes which are involved in RNA biosynthesis. And then there were genes which were involved in DNA replication, mitotic spindle formation. So these genes were related to mitotic cell division. Okay, so I was just thinking like what is happening? Why? Because these functions are not known for TMPRSS2. And I was thinking, let's say virus infect a particular cell, and if it is actually proliferating, so basically it is going to divide, its cytoplasm is totally disturbed. Not disturbed, but it is not like normal cells. So what will happen to viral replication? Would it still replicate? Considering that the TMPRSS2 may help RNA genome replication. So still, I don't have any idea, but this is very interesting thing, because the 9 genes out of 9 genes are actually involved in negative regulation of cell cycle phase transition. So my speculation is maybe when TMPRSS2 goes down, the cell cycle phases are restricted, and then cells are not dividing, and then virus can happily replicate in them. And the genes which were associated with both ACE2 and TMPRSS2 TMPRSS2 receptor, they are again involved in, most of them are involved in cell cycle and phosphorus metabolism. I also see nitrogen metabolism. So these metabolic processes, they are required to synthesize DNA or protein molecules. And also some interesting terms were response to hypoxia or leukocyte migration. So response to hypoxia is interesting term, because in many COVID patients, they have this breathing problem. So then I wanted to know if these genes are really involved in somehow in infection. they must be, some of them must be interacting with SARS-CoV-2 protein. So I collected 300 proteins, which were screened from pull down assays with SARS-CoV-2 proteins. And out of these 332, 38 genes are already exist in my list. So it seems, okay, maybe these are the functions which virus is using to accomplish its reproduction inside cell. And what was interesting is one of the protein is poliovirus receptor, okay? And which is here, and basically most of these genes they are involving ear-related functions, glycosylations, and some phospholipid metabolism. So I will talk about this polio receptor later, but this is very interesting because poliovirus is also RNA virus. So since these results were from large-scale microarray data analysis from human, I also wanted to see if these results are consistent. I find these results consistent in mouse also. Okay. So I just want to mention that coronavirus cannot infect mouse because there are some changes in ACE2 receptor, so coronavirus cannot bind to mice membranes and form attachment site. But if you replace this mouse ACE2 enzyme with human counterpart, then these mice get active infection. It means that the downstream processes, ACE2 downstream processes actually concerned in human and mouse. So I can use this data. So for that, for some other purpose, I actually wanted to have the data set from all over body. And I eventually found one data set which was from 20 different organs of mouse and containing approximately 45,000 single cells. So I divided these single cells into two groups. One, which they are expressing both ACE2 and TMPRSS2 and the all other remaining cells. So basically, I'm actually trying to mimic in silico knockdown, let's say. So we have ACE2 and TMPRSS2, both double positive cells, and one of them is not positive in the other group. So then I performed differential expression analysis. And here what you see is the result for the genes which were co-expressed with ACE2 and TMPRSS2. They have one-fold log-fold change expression in ACE2 and TMPRSS2 positive cells, and very rarely these genes were down-regulated in these cells. Okay, so these are upregulated. So these genes are upregulated, and here we can say that in mouse as well as in human, these genes are actually dependent on ACE2 and TMPRSS2. So I try to investigate these genes and actually some other genes because of some other purpose. But right now, what is important here is that of course, ACE2 and TMPRSS2 were highly expressed. But then I also found TMPRSS4, which is a homologue of TMPRSS2, which is also massively upregulated. And then I also see here vitamin D receptor. Okay, so and PVR. So vitamin D receptor was very strongly correlated with ACE2. So I selected these three genes to study them further. Okay. One more thing I would like to point out here: there were many genes expressing keratin proteins were upregulated. And these keratins, they are produced by keratinocytes, which are skin cells. I mean, this found in a lot of organs, but mainly in skin and tongue cells. So summary of my investigation so far is I have used ACE2 and TMPRSS2 genes to identify approximately 1200 genes which may work with them, and these genes were significantly downregulated in all 3 coronavirus infections. And mostly these genes were related to immune response. I did talk about this neutrophil degranulation, cell cycle, and virus-related processes. And the striking thing about TMPRSS2, it seems it is involved in somehow involved in RNA regulation of RNA biosynthesis and mitotic cell division, and with some other immune cell-related metabolic processes. And 38 genes co-expressed genes physically interact with SARS-CoV-2 protein, and this could be good starting point to study further. So I selected these 3 genes to study them further. So this is TMPRSS4. Okay. So briefly, I will talk about these 2 genes, and I will go in deep with into TMPRSS4 functions. So this is the statistics using two different statistical methods. I found that TMPRSS4 and VDR, they were among the topmost genes associated with ACE2 and TMPRSS2 positive cells. And this was not in that range, but still I considered because this protein physically interact with SARS-CoV-2 protein. And I think these are potential therapeutic target genes because poliovirus receptor expression can be modulated with poliovirus vaccine. And this vitamin D receptor functions can be modulated by vitamin D supplements. And TMPRSS2, I'm going to talk in more detail about this, but there are some drugs, they are not under clinical trial, but they inhibit TMPRSS4 functions. So maybe those molecules can be used to find some more drugs to either inhibit or accelerate the TMPRSS4 expression. So there was one article, basically this is published in Science, and this article discusses what are the possibilities of different existing live vaccines which can be used to treat COVID-19 patients. Okay. And they focused in this article about poliovirus vaccine, but the arguments were entirely different, and the argument was when we take vaccine, actually it mounts immune response, and it may help to also protect from other diseases. I'm not sure if it works, but still, since now I find this poliovirus receptor here, it may work like that. So there are some reasons to think like coronavirus can be a potential target. So first, obvious is because I identified. Second, this physically interact with SARS-CoV protein. But the interesting thing, it also interact with other proteins too. So this actually indicate that there are common themes which are used by these viruses, okay? But we haven't explored this so far. And ORF8 actually has endoplasmic reticulum targeting sequence. So it means it exported to endoplasmic reticulum, and its interacting proteins, actually they are involving ER protein quality control. Furthermore, there are some symptoms which are common in poliovirus, but I think that is not big. What surprises me and what made me think about this as a good therapeutic target is, if you look at children's, in current and previous two pandemics, they rarely get this infection. And I believe they have taken doses of polioviruses very recently, and that immunity still is very strong and maybe protecting them from coronavirus infections. But of course, this is striking to see that poliovirus actually interact with SARS-CoV-2 protein. Then vitamin D receptors. So there are compelling evidences suggest that the levels of vitamin D correlates with the severity of the disease. But this is very recent editorial paper which says, okay, so benefits are possible, but where is the evidence? Because all of them are sparse, indirect, or inconclusive. Okay? So we don't have any molecular level evidence as of now. Okay? So in general, the vitamin D receptor functions are associated with calcium homeostasis. So when vitamin D binds to this receptor, it activates vitamin D responsive genes. Okay? And it has been found that the expression of vitamin D receptor decreases with the age. So I checked some data on vitamin D, and I found some very interesting aspects about vitamin D and its level. So first, I'm gonna just explain this curve. Okay. So these are the deaths happened in last 25 years in USA, and these are the days of the year. So you can assume that this is January to December. Okay, and the number of deaths, okay, they decline during summer months, okay, and they again spike up during at the beginning of the winter. Okay? And this is from different papers. And then there is a data about vitamin D levels. Actually, this data exists in a number of different publications, but I just selected this one. So if you look at the vitamin D levels in different seasons, so they drop down in January, and then they basically picks up during summer, and again they go down during winter. And this is expected because vitamin D is synthesized by sunlight, and during winter months we don't have that much sunlight in north part of the globe. So basically this the seasonal death rate in USA, it anti-correlates with the levels of vitamin D. And if you look at what caused most of these deaths, okay, and it was because of respiratory, cardiovascular, digestive system related disorders. And all these three symptoms are very prominent feature of SARS-CoV-2 infection. And then I checked cases of number of cases every month from the beginning of pandemic and until the March. Okay, so this data is we don't have this data because the WHO started to count after they declared the pandemic, and since then you can see that the numbers have been increasing. So there could be a lot of reasons, but what is interesting is that again they are declining, and I hope that by June those numbers will be even more or less. If you look at the number of cases worldwide, okay, so it reveals very interesting thing. So for example, if you look at the sub-Saharan South Africa or India or this part of world, so the number of cases, okay, per 1,000 population, they are less compared to people who are in Europe or those who share ancestry. Because if you look at the evolutionary path, so first human migrated from Botswana, maybe in Arabian Peninsula, and they are more likely to migrate to Europe, and then from Europe they migrated to Russia, and from Russia they enter into USA, and the South American population is actually a mixture of European population. So it seems the temperature actually has impact on the severity of COVID patients, okay? and the populations of Americas, they are more susceptible than the African, sub-Saharan African, or Southeast Asian people. So one more evidence suggests that there are two more papers which shows that vitamin D levels decreases with age, and also there is another paper which shows that people who have vitamin D levels more than 380 nanogram per ml in serum, okay, those people, they are more likely to not develop viral infections. So now I'm going to quickly talk about this transmembrane protein, protease serine 4, because I think this is very underrated gene in COVID pathogenesis. So when the first time they identified TMPRSS2 as a receptor for SARS-CoV-2, they also did some experiment with this gene, and they found that this gene actually can allow a membrane fusion of SARS-CoV-2 as good as the TMPRSS2. Okay, but the molecular mechanism behind the activation of S protein is not known. So they just focused on this gene, and this gene was left out. So now currently there is another paper which was published very recently, and it suggests that in intestine, TMPRSS2 and TMPRSS4, they promote SARS-CoV-2 replication in intestinal enterocytes. Again, this paper was also not in light, but I think this was very interesting discovery. So if you look at the domain organization of TMPRSS2 and TMPRSS4, their extracellular domain is almost very similar. All the active sites and which require for this extracellular region, they are present in both of these proteins. Okay, so my assumption was they must have identical or equivalent function, at least with these extracellular domains. So I wanted to study them, so I did some sequence analysis, and I found very interesting results. So their structural homologs were mainly proteins which are involved in coagulation, fibrinolysis, and alternative complement pathway, which are involved in immune response. And then then I tried to study their domain organization, and I found another interesting thing. So the blood coagulation related functions of all of homologs of all these proteins, they are performed by a protease domain which belong to high temperature requirement A protein family. And this family, so I have studied this family, the proteins which contain this domain in my previous study. And what it seems like, these proteins, these domains, they are prototype of enzymes or proteases which are involved in protein quality response, including temperature. So null mutant of STRA gene in E. coli, they don't survive at high temperature. So the question is why all these genes have this particular domain. For me, it seems like it has something to do with temperature resistant. So I predicted TMPRSS2 and TMPRSS4 structures, and I also checked if the protease domain can be inhibited or is similar to known blood clot resolving proteins, for example, plasminogen. And I found that the overlap was very striking, and especially the active site of these proteases, they perfectly match for TMPRSS2 and TMPRSS4 as well. So I'm just going to skip this slide, but the thing is that I think the European population or South Americas, they must have genes which actually have a selection pressure of temperature. So then I wanted to study whether these genes their expression correlates in different tissues and cell types. Okay, so this is very busy slide, but just I just want to say, like in mouse, so these are like 50,000 cells from 20 different organs. So what I see that AC2 is not highly correlated with TMPRSS2. Actually, AC2 is much better correlated with TMPRSS4, and TMPRSS2 and TMPRSS4, they are highly correlated in a lot of different organs. Okay, so AC2 and TMPRSS2, they are mainly co-expressed in intestine, bladder, and skin cells. And AC2 and TMPRSS4, they are co-expressed in tongue, skin, and also intestine. So then I wanted to see their expression pattern in human tissue. And I see almost same results, but now the things are very different here. For example, in lung, AC2 and TMPRSS2 are highly co-expressed. Now, one possible reason for these differences, the human tissue data comes from RNA-seq, and it is a mixture of all cells. So if expression signal is mixed, okay, and in single cells you have straight signal from cells. Okay. So maybe this AC2 and TMPRSS4, they are expressed on two different membranes, two different cell types in tissue, but still here you see this signal in unified manner. Actually these two results, even if they look contradictory, they have one significant aspect. SARS-CoV-2 can be activated, can bind to AC2 receptor on one cell and activated by TMPRSS2 receptor on the other cell. So this shows like AC2. So it means SARS-CoV-2 actually infect a lot of different cell types, almost all organs. So I didn't show the data, but what I found that the whole gastrointestinal tract shows co-expression of all these genes. And then I wanted to know which cells actually express combination of these cells. And strikingly, what I found is that most of these cells are actually from large intestine and also from tongue, keratinocyte. And then, so this is just to show for only two organs. So large intestine, and this is the expression of different cells, and these are the number of cells in this organ. Okay. So large intestine is highly susceptible site for all of for coronavirus. Okay. And I also feel that the tongue is actually another aspect people have missed so far. And there are symptoms which are like decoloration of tongue have been found in COVID patient, and also the skin rash or skin problems, they can be explained by the presence of these receptors on skin. And diarrhea is one of the most common symptom of all COVID patients, and it seems it may be because of this, because these receptors, they are expressed in very high amount on the different cell types in intestine. So I confirmed these findings also in almost more than 100 single cell transcriptomic data sets which were available. And again, I find that the main site of primary site of SARS-CoV infection seems to be intestine and not lung, because I don't see a lot of convincing data to suggest that, as we believe. So I have already discussed most of these conclusions, and I think I'm running out of time. So I'm just gonna just wind up here. So basically, I think these genes, they are very critical part of innate immune response, okay? And their down regulation is actually required so that these viruses can easily enter into cell and replicate and infect and develop into disease. Okay, so that's it. I'm done, and I would like to acknowledge the data sources from where I got all. I have used all publicly available data. So I got tissue specific tissue gene expression data from GTX, and then those 1,000 microarrays from GEO, and the tabula modis, they provide data for mouse organs. I would like to acknowledge Professor Alfredo Varela for critically reviewing this work, and Amit Singh from Austria. He did a lot of analysis which actually I didn't present, but basically he did prediction. He did molecular docking analysis of different blood clotting inhibitors on the TMPRSS4 and TMPRSS2 predicted structures. And Esteban, he helped me to analyze some human single cell RNA, single cell transcriptomic data. So thank you very much for your kind attention, and I would like to answer some questions if you have.
Thank you very much, Vijay. Very interesting seminar. We already have several written questions. I'm going to read them. Carlos Lozano is that he has an excellent seminar. He says congratulations.
Thank you.
Then Dr. Isabel Mendez has two questions. The first one is, among enriched genes in response to hormone, are there sexual hormones?
Actually, I haven't seen which hormonal genes are there. Okay, but the TMPRSS2 gene is induced by androgen, so I expect there are some genes, and but for sure I know there are some steroid hormone genes involved in steroid hormone pathway, but I haven't checked specifically those genes.
Okay, the second question is, did you find some correlation with adaptive immune response genes?
No, as of now, I have only seen innate immune response, but I must mention AC2, some of the AC2 genes, okay, they seem to be involved in antigen presentation and processing. So yeah, again, I need, so maybe there are some genes, but not much. So mostly it is innate immune response, I think.
Okay, Juan Carlo Olais says, considering your correlations, we see that there are many organ histologic targets that can be infected. I even read that SARS-CoV-2 was reported in cerebrospinal fluid. Has this been confirmed?
Okay, so there is one gene. I don't know its name, but it is NRP1. So this is specifically expressed on neuronal cells, and there is one Science or Nature paper which showed experimentally that this receptor allow SARS-CoV-2 entry into neurons. In olfactory neurons, for sure, we know SARS-CoV-2 can enter. Infection, I don't know.
Okay, Juan Carlos also says a great presentation, by the way. And then Dr. Fernando Peña asked, could you elaborate a bit more about the genes you identified related to hypoxic response? Any interesting or famous one?
Okay, so one gene I can mention, and that is already known, this hypoxia inducible factor. That gene is there for sure, but other genes I don't know. One more thing, there are genes which are involved in mitochondrial organization, which were enriched in the list, which were associated with TMPRSS2. So cytochrome, yeah, I don't remember, but there are some genes. Yeah, they are present in the data.
Okay, Juan Carlos says thank you. And let me see if someone wants to talk by the microphone, but not so far. Dr. Alfredo Varela asked, do you think the link of TMPRSS2 and TMPRSS4 to fibrinolysis could explain the results that are obtained in the clinic treating COVID-19 with anticoagulants?
Yeah, those results, yeah, they can be explained by this, by these results. Yeah, because we clearly see the protease domain is enough for either blood clotting or even for blood clot resolution, and these genes they are mixed. They are either fibrinolysis related and also coagulation related. But there is evidence that one enzyme can perform both functions. For me, it is more likely TMPRSS2 and TMPRSS4 are involved in blood clotting resolve, because the active site, predicted structure and active site, they perfectly match with plasminogen, and which is known protein involved in blood clotting resolution.
Okay. Fernando Peña asked, regarding HIV association, is it HIV1 or HIV2?
I guess he's asking me if it is STRA1 or 2.
There is about isoforms of HIV, this transcriptional factor. HIV1 or HIV2?
HIV, hypoxia inducible factor, it was 1.
It's 1, okay. No more questions. No one wants to use a microphone. Second question from Dr. Varela's. Could the role of TMPRSS2 and 4 be related to the blood clotting problems observed in some patients with the Johnson & Johnson and AstraZeneca vaccines?
Actually, I didn't get the question. Mauricio, I didn't get the question.
Let me read again. Could the role of TMPRSS2 and 4 be related to the blood clotting problems observed in some patients with the Johnson & Johnson and AstraZeneca vaccines?
That I'm not sure. I don't know.
Okay, and Dr. Sofia Diaz says, congratulations, very good seminar. Thank you.
Thank you so much.
Let me check. No more written questions, and no one wants to use the microphone. Then, Vijay, thank you very much for your very illustrative seminar. And gracias a todos. Nos vemos la siguiente semana. Thank you, Vijay.
Thank you so much for attending.